You know that MRD has been traditionally viewed as a prognostic factor, and it is one of the most relevant prognostic factors in myeloma. More recently, it has also been investigated as a tool to guide on specific treatment decisions, and these investigations obviously taking place in clinical trials. But there is a third potential application of MRD that remained unexplored until now. That is its utility to accelerate drug development and the approval of new drugs and new regimens in the context of using MRD as a surrogate endpoint in clinical trials. Now, this is a different, how can I say, a different view of traditional MRD. It's not that MRD negative patients will do better than MRD positive patients. That is well established. It's not about the methods, the sensitivity. No, it's about predicting the effect on treatment in traditional endpoints such as progression-free and overall survival based on the effect of treatment in different MRD negative rates in randomized clinical trials. In other words, to predict what would be the outcome of a large and long clinical trial 6 or 12 months after the last patient was enrolled, anticipating the readout many years before the readout based on progression-free and overall survival. If we are able to show this, then a conditional approval based on MRD surrogacy could imply that drugs would be available to patients much faster. Now, this research about MRD surrogacy is taking place within the I-Squared team initiative that involves many different academic centers and even pharmaceutical companies as well, that relies on the individual patient level data. That is the real deal. What we present here at EHA is a simultaneous parallel analysis based on published data where we looked for the correlation between different MRD negative rates and the hazard ratio in PFS on randomized clinical trials using flow and sequencing methods to monitor MRD. And the results look promising. It's not an ideal correlation, it's not a perfect correlation, but there is a significant correlation, and I think that it is a value of around 0.6, 0.7, depending on the trials that you select and the inclusion criteria, that makes us believe that the effort is worthy. We know it is because it would imply having drugs faster to the patients, and that there are some chances that MRD can become a surrogate endpoint to be used in clinical trials.